And the first part that we're going to look at is how the brain is protected from damage. We're going to do this at quite a crude level, and we're going to cover this because it relates to the next part, which is looking at how the spinal cord functions, but also how the spinal cord is protected from damage. And there's a lot of similarity between the two, as the meninges are in fact connected between the brain and the spinal cord. So, like I said, the brain itself is protected from mechanical forces by numerous protective measures. You've got the cranial bones themselves, you've got the cranial meninges, and of course you've got the cerebrospinal fluid. The cranial meninges themselves, the layers are continuous with the spinal meninges. In other words, the three layers, you've got the dura mater, the arachnoid mater, and the pia mater. And these are just shown in this image below now. So you can see on the outside we have the cranium, the skull, or the cranial bones. You then have the dura space. So we're now into the meninges. You have the dura mater, you have the dural sinus, and then you have the inner layer, the dura of the dura mater. You then have the subdural space in between them before moving on to the arachnoid mater, where you have the different areas, the arachnoid membrane, the subarachnoid space, and the arachnoid trabeculae, before finally hitting on to the pia mater. And that's bound to the surface of the brain by astrocytes, which have very important roles within the brain itself. And the dura mater itself, of course, on the outer layer, that's attached or fused to the cranial bones. So the cranial bones themselves are obvious. They provide that protection, that solid structure, a bit like the outside of a car provides the support to us. So you've got you've got a car with its structure, and you've got a seat belt to help in that protection. And it's the same thing with the brain. You've got the outer structure, which is the bone, and then you've got the cranial meninges, which are kind of acting a little bit like a seat belt. So when we're looking at these, the outer and inner dura layers, so remember these are in this region here, these are separated by a gap containing fluids and blood vessels. That is our dural sinuses, as is shown in the image itself. And the veins of the brain open up into the dural sinuses, delivering blood onto the jugular veins. And the arachnoid mater, the next one down, that covers the brain, and it helps to provide a smooth surface that does not follow the brain's folds, which is in contrast to the pia mater itself, which sticks to every fold of the brain's surface. And like I said, it's anchored to those by the astrocytes themselves. So, carrying on with that, well, so what is the cranial meninges doing? Well, they're obviously involved in protection. The cranial bones provide that mechanical protection by cradling the brain, and that has impacts in itself, of course, because the cranial bones can also provide a threat because they can break, and that's countered by the cranial meninges and the CSF. So if your skull does snap or break or splinter, you've got the cranial meninges there and the CSF helping to provide that support. The dura acts like a seatbelt, holding that brain in position, and the CSF in the subarachnoid space acts like a bumper, helping to cushion against sudden jolts. So that's the kind of an easy way to think about how that works. You've got the bones acting as a car itself, you've got the CSF is a bit like a bumper, and then the dura is acting like a seat belt itself. Give you just a little indication of how many cranial traumas there are. So I haven't got figures for the UK or Australia, but in America there are about 8 million cranial traumas every year, about 1 in 8 of which is serious. And that's in part because only 1 in 8 is serious, and that's in part because of the CSF and those dural folds helping with that protection. And then we've got the CSF, or the cerebrospinal fluid. Now that surrounds and bathes the exposed surfaces of the CNS, and it has got many, many different functions. As we've talked about, it helps to cushion against damage. It helps to support the brain. So the supporting role of CSF, in essence, the brain is suspended inside the cranium and floats within the CSF, if we want to think about it in a simple way. It helps to transport nutrients, chemical messengers, and waste products. So it's in constant communication with the interstitial fluid that's surrounding the neurons of the CNS. But have a little bit of a guess. What is the CSF volume? What is the volume of cerebrospinal fluid within your body right now? And how much do you think you make each day? Because it's always getting replaced, of course. So it's about 150 mil and 500 mils. So you've got about 150 mil floating around inside you, and you make around about 500 mils. But what its composition is it the same as plasma, or is it different to plasma? So if you're looking at plasma, you've got the osmolarity, at 290, pH 7.4, glucose about 5.5, cholesterol about 4.5, total cholesterol, and you've got about 60 grams per litre of protein. What about the CSF? Is that different? Is that the same? Does it help if it's different or the same for people who might have various conditions such as meningitis? Do you get a difference between bacterial meningitis and viral meningitis, for example? So if we're looking at the osmolarity is about the same. It's at about 290 milliosmoles per litre. The pH, that's a little bit lower, 7.33. The pH is thought to be a bit lower because of the increased amount of CO2 is lower due to the increased amount of CO2. And you've got glucose. The glucose CS levels are lower as well than in the plasma. Cholesterol is very much lower, and the protein is also very much lower as well. So you can see there are significant differences between plasma and CSF values. So you can actually, if someone has meningitis, you can take a CSF sample and actually take a measure of glucose, for example, to help distinguish whether it's bacterial or viral, because the glucose levels are lower in bacterial meningitis as a rule than in viral meningitis. So now we're going to look at the structure and function of the spinal cord. So the spinal cord itself is about 45 centimeters long. It has a max width of about 14 millimeters. And it does have a couple of enlargements in a couple of places. So if we're looking at the image, you can see the cervical enlargement. And you can also see further down, you can see the lumbar enlargement as well. And we'll talk about why they're there in a second. And when we're looking at a cross-sectional view of it, so we know that we've got the cervical spinal nerves, the thoracic spinal nerves, the lumbar, and then the sacral spinal nerves. And if we're taking a snapshot through it, we're taking a nice little look, and you can see the grey matter, which is in that nice butterfly shape, and you can say the white matter. And obviously grey matter, simply put, is computer, and white matter is cables. And the posterior median sulcus, as we can already see, that helps to, that has a shallow groove in it. That has a shallow groove. You've got the anterior median fissure, has a deeper groove along the anterior or the ventral surface. And of course, the grey matter, as we've already talked about, is greatest in areas dedicated to sensory and motor information. In particular, we're thinking about the limbs because of the large amount of nerves going to them. So you've got the cervical enlargement, looking at the shoulder and upper limbs, and you've got the lumbar enlargement, the pelvis, and the lower limbs. So now if we take a snapshot and have a look at it, you can see that every spinal segment is associated with a pair of dorsal root ganglia near the spinal cord. And so the dorsal root ganglia is just an area where the cell bodies of the sensory neurons accumulate. So the afferent sensory neurons, they come in through the spinal nerve and then enter through the dorsal root with their cell bodies in the dorsal root neuron before they synapse in the grey matter in the dorsal horn. The efferent motor neurons come out through the front, so they're coming out through the ventral root and then go through the spinal nerve again, just like the sensory neurons, and then before going to their final destination. The grey matter, as you'll know, consists of the neuronal cell bodies, dendrites and interneurons, the white matter, the myelinated fibers. So while the grey matter is primarily associated with processing and cognition, the white matter really modulates the distribution of action potentials, acting as a relay and coordinating communication between different brain regions. And distal to each dorsal root ganglion, the sensory and motor neurons are bound into a single spinal nerve, as I've already mentioned, which is about 1 centimeter long or so. So now the spinal meninges, and these flow on from the cranial meninges that we've already talked about. So they surround the spinal cord and they help to provide physical stability and shock absorption. The spinal meninges are continuous with the cranial meninges, and just like in those, you've got the outer dura mater, you've got the middle arachnoid mater, and you've got the inner pia mater. And there you can see there's the spinal nerve, here's your ventral root, and here's your dorsal root, and then the grey and the white matter of the spinal cord itself. So this gives you a nice simplified image of the structure of the spinal cord itself. Blood vessels branching within these layers help to deliver nutrients to the spinal cord. And of course, we all know what meningitis is. Meningitis is inflammation of the meningeal membranes. This can be by virus or bacterial, as we've already talked about, and it can be spinal, cerebral, or both as well. And of course, meningitis is dangerous because it can disrupt normal circulatory and CSF supplies, damaging and killing neurons in such affected areas. So looking at the spinal meninges again in a little bit more detail this time, here again you can see the outer dura mater, then the epidural space, which is not present in the cranial meninges, the inner dura mater, the arachnoid membrane, the subarachnoid space, which has the CSF, the pia mater, and then the spinal cord, of course. So in this case, the epidural space, of course, that is a needle location for an epidural block. For example, lumbar puncture, it goes into the subarachnoid space. So, and as we just said, anesthetics are often injected into this space where they therefore only affect nerves in that particular vicinity. It's called an epidural block. So you get a temporary loss of sensory or motor pathways, depending on where the region is. Sensory and motor pathways. So the pia mater, that is thin and it's attached to the spinal cord. You've got the arachnoid, which forms the blood CSF barrier. Remember, the CSF is a nice shock absorber and a good diffusion medium for gases, nutrients, chemical messengers, and waste products. And then you've got the dura mater, which is two thick layers with the epidural space in between. And so that's where you have your fats and your blood vessels. And the adipose in the dura provides that protective padding. The capillaries servicing the spinal cord, they run along the surface of the pia mater within the subarachnoid space itself. So a bit of a question for you, or three questions as the case may be. So press pause, read the questions, then give us the answers. Okay, so name the three spinal meninges. Well, it's nice and easy. Dura, arachnoid, and pia. Damage to which root of a spinal nerve would interfere with motor function? So hopefully you put a ventral root, which can be visceral or somatic motor fibers. And then where is CSF that surrounds the spinal cord located? That's in your subarachnoid space, so beneath the arachnoid mater and superficial to the pia mater. So now we're going to look at the grey and white matter in a little bit more detail within the spinal cord itself. So we've already mentioned the anterior median fissure and the posterior median sulcus. So the anterior median fissure and median sulcus, they mark the division between the left and right sides of the spinal cord. And the white matter contains myelinated and unmyelinated axons, and the grey matter forms this butterfly shape, and it contains the cell bodies and unmyelinated axons. The projections of grey matter towards the outer surface are called horns, and this is what we're showing here. So we have the dorsal horn and we have the ventral horn, and they contain specific nuclei, so visceral and somatic and sensory and motor. So again, here's a nice little image. So we have our sensory neurons. They come in through the dorsal root, and there's our dorsal root ganglion where the cell body is. Comes in and they synapse in the dorsal horn. And if it's somatic, it goes there, so your skeletal muscle, for example. If it's visceral, so your internal organs, it'll synapse at this point. And then the ventral runs, again, you can see you've got the motor nuclei. You can see there's your somatic and there's your visceral points. So that's present. This is your ventral horn where you've got your somatic motor nuclei. So cell bodies of neurons in the CNS grey matter are organized into those functional groups called nuclei, and they're motor and sensory nuclei, just groups of cell bodies. The dorsal grey horns, as we've talked about, contain the somatic and visceral sensory nuclei. So there we go, showing us very nicely in this point. So they're receiving that sensory information coming in. Whereas the anterior grey horn contains the somatic motor nuclei. So now we'll go on to the white matter organization. And it's divided into three main regions, or columns, or funiculi. So the posterior white columns, which are present up at this point, they are between the posterior grey horns, which is there, and the posterior median sulcus, which is there. So that's your posterior white column. So this is the region where we're going to be carrying information up and down the spinal cord. We've got then the anterior white columns, and this is present between the anterior grey horn and the anterior median fissure. And as you might expect, we also have the lateral white columns, and this is between the anterior and posterior columns. So these columns, they contain tracts, or basically bundles of axons, which share functional and structural characteristics. So in other words, they have very similar diameters or myelination and therefore conduction speeds. And that column bundles with those relatively uniform speeds tend to relay the same type of information, so whether it's sensory or motor, in the same directions. And these columns obviously have axons going up and down the spinal cord to and from the brain. So what are some of these columns? So this is all that this image is showing. It's just showing you some of the ascending tracks, so some of them that are going upwards, so from the spine to the cerebellum, for example, if we're thinking about this ventral spinocerebellar tract. Or lateral spinothalamic, it's going from the spine to the thalamus and then beyond into the cerebral cortex. So they're kind of, at least they aid in some way to help you locate or determine its location from their very name. Same thing here, lateral corticospinal, going from the cortex down to the spine. So it's kind of helping you understand where they might be going in some ways. So just as a couple of examples for you, and so that you know, they are multi-neuron pathways. And we'll talk about first, second, and third order neurons in a minute, the ascending tracts. They connect the brain to the body periphery, and each tract is made up of axons with similar destinations as functions, as we've already said. So some examples, you've got your dorsal white column. That's involved in fine touch sensation, and it goes via the thalamus to the cerebral cortex, the somatosensory cortex. You've got the spinocerebellar tracts, and as you might expect, they provide proprioceptive information and in these cases, the lower limb and the trunk. So they go up to the cerebellum and give that proprioceptive information to talk about where are my joints in space, how can I perform coordinated movements, because now I know where my joints are in space. You've got lateral spinothalamic tract, which is involved in pain and temperature sensation. And you've got ventral spinothalamic tract, which is to do with crude touch as opposed to fine touch. So, thinking about those ascending pathways, they usually have chains of 3 successive neurons to the various brain areas. Not always 3, because the cerebellum you just have the 2, but they usually have 3 successive neurons. So the first is, this is an example that we're going to look at now. So we're looking at the lateral spinothalamic pathway. Remember that transmits pain and temperature. And we're presuming that we're lighting a match looking at this image onto the bottom of someone's foot. So we're stimulating temperature and pain, I guess. It's stimulating both. So the first order neurons, the cell body lies in the dorsal root ganglion. So remember, you have your sensory neuron comes in through the spinal nerve, up through the dorsal root. The cell body is in the dorsal root ganglion, and then it comes in and synapses at the dorsal horn rather. And it's helping to conduct impulses from skin and proprioceptors, etc. So as a general, if we're thinking about first order neurons. So it's helping to transmit the somatosensory information through the spinal nerve and dorsal root to the dorsal horn, as we've just talked about, of the spinal cord. It's then going to obviously cross over and make its way up the spinal cord itself. It's also important to realize that cell bodies from the face, for example, they lie in the cranial ganglion, and impulses are transmitted via cranial nerves. So just that you have a concept that not everything goes through the spinal cord, as you would imagine. So the second order neurons, so we've dealt with the first order neuron. Remember, this receives the sensory information and takes it to the dorsal horn, where it synapses in the dorsal horn with the second order neurons. And so the cell bodies in this case, they lie in the dorsal horn. So the cell bodies of the first order neuron are in the ganglion. Second order neurons, the cell bodies, are in the dorsal horn of the spinal cord. They cross over and they transmit the information up the spinal cord towards the thalamus or cerebellum if we're thinking about proprioceptive information. Then we move on to the third order neurons. And this is where the cell bodies are now present in the thalamus. So this is, in this example, remember we're looking at the spinothalamic pathway. It's coming up through this, it's passing up through this brainstem, comes up to the thalamus where it synapses. And here we've got the third order neuron. And this is the cell body of the third order neuron is present within the thalamus. and it relays information to the primary somatosensory cortex of the cerebral cortex, because we're thinking about pain and temperature. As I've already mentioned, there are no third-order neurons at the cerebellum because the information going to the cerebellum doesn't synapse within the thalamus itself. And so in this case, with the third-order neurons, so I should have mentioned this already, remember when it gets up to the primary somatosensory cortex and then gets on to the association areas, that's when the information starts to get processed and interpreted. So remembering what the thalamus does, it relays the information, but it also helps to filter out irrelevant stuff that we don't really need to know about. Okay, so it has a filtering role as well as transmission role to the correct place. Okay, so what about the descending pathways and tracks? So we know that the ascending pathways have first-order neurons, second-order neurons, and sometimes third-order neurons. And in the descending pathways, we're thinking about upper motor neurons and lower motor neurons. The upper motor neurons are also called pyramidal cells. This is shown up at the top for you, just so you know the names. So upper motor neurons, pyramidal cells, and then you've got lower motor neurons, also called somatic motor neurons, just so that you're aware. And in this case, you can see that we're looking at two different tracks. We're looking at the ventral corticospinal tract, and we've also got the lateral corticospinal tract. They're both going from the cortex to the spine, but if you're looking at them, you can see that they cross in different places. So we all know that they cross from one side to the other. They're just crossing in different places. So you've got decussation of the pyramids going on there. So they help to deliver impulses from the brain to the spinal cord and involve both the upper and lower motor neurons, as we've already talked about. The upper motor neurons originate in the primary motor cortex, and they send impulses through the brainstem via the various corticospinal tracts. At the spinal cord, the upper motor neurons synapse with the interneurons or the lower motor neurons, and then we get stimulation of lower motor neurons activates the skeletal muscle with which they are associated. So nice and simple. So a nice little question for you to finish off on. So which of the following are correct regarding white matter tracks? The cell bodies of peripheral nervous system neurons lie in nuclei. That is obviously incorrect. The cell bodies of CNS neurons lie in nuclei. The PNS ones lie in ganglia. First-order neuron cell bodies of ascending tracts lie in the dorsal root ganglion. That is correct. Second-order neurons of ascending tracts transmit impulses to the thalamus or to the cerebral cortex. That's incorrect. They either transmit to the thalamus or to the cerebellum. It's the third-order neuron that takes it from the thalamus to the cerebral cortex. Descending tracts deliver impulses from the brain to the spinal cord. It's obviously correct.